Yeah, I think people are joining now. The number is increasing. Let's wait just 30 seconds. Hello, everyone. Welcome to the cannabis cultivation webinar. We will be starting shortly, but first, we would like to allow a brief moment for attendees to join the webinar. The number is increasing, so again, we will just wait for just 10 seconds, then we can get started. Maybe another 10 seconds. All right. Once again, welcome to the July Cannabis Cultivation webinar. It seems like the number of attendees has increased and stabilized, so I can begin now. Hello, my name is Sang Park, a senior scientist at the ICR and chair of this webinar series. The ICR and Leibniz Institute in Israel are pleased to have you join us for the July webinar. Before we begin our presentation, I would like to inform you that ICR will be hosting the ICR Cannabis Research Conference in Denver, Colorado from August 3rd through 5th. If you are interested in participating, please visit the conference homepage or contact us directly, and we will be happy to provide you with more information. Also, I would like to provide some information regarding the logistics of the webinar. Today's presentation will be followed by a short question and answer period. To ask a question, please use the Q&A feature in the Zoom toolbar. You can enter questions at any time during the presentation, and we will address as many questions as time allows until the end of the hour. Please do not use the chat function to pose questions for the Q&A period. Instead, please reserve the chat function for technical issues and questions. Both the Q&A and chat functions can be found in the toolbar on your Zoom screen. It is now my pleasure to introduce Dr. Nirit Bernstein, Senior Research Scientist at the Volcanic Center, Ministry of Agriculture, Israel, and co-chair of this webinar series. She will be introducing today's presenter and moderating the webinar. Nirit.
Thank you, Sang. It's my first of all, welcome all the people that are joining us wherever you are from, and it's my honor to introduce Professor Frederik "Fred" Stevens, today's speaker. Professor Stevens earned his master's in pharmacy in 1988 and a Pharm.D in 1990 and Ph.D. in medicinal chemistry in 1995 from the University of Groningen, the Netherlands. He received postdoctoral training at Oregon State University, at the Free University of Amsterdam, and at the Leibniz Institute of Plant Biochemistry in Germany. And in 2002, he joined the faculty at Oregon State University in Corvallis, where he holds positions today as Professor of Pharmacology Sciences and Associate Dean for Research in the College of Pharmacy. Professor Stevens has authored more than 175 articles in peer-reviewed journals. He received 5 awards for excellence in teaching at the Oregon State University College of Pharmacy. He holds a guest professorship at the University of Antwerp in Belgium, where he also teaches a course on medicinal natural products. Professor Stevens serves on the executive editorial board of molecular nutrition and food research, and has served the Phytochemical Society in North America as president. His research, which is funded by the National Institute of Health and the US Department of Agriculture, aims to determine the role and function of dietary phytochemicals in human health and diseases. Mass spectrometry-based metabolomics is a major research tool in Professor Stevens' lab for the discovery of biological effects of vitamins and phytochemicals in cultured cells, animal models and disease, and in humans. And we are very grateful to Professor Stevens for spending the time, taking the time today to talk to us about chemistry and pharmacology of cannabinoids.
Thank you, Dr. Bernstein, for this introduction. I will get started, otherwise I run out of time. So today I'm going to give a little, I don't really know what to expect from the audience, so I will start at a very basic level, and then gradually I will get into our research from our laboratory. So I'm actually from Oregon State University, and at this university we have a global hemp innovation center. It is mentioned on the website that you can visit yourself. It was founded in 2018, and the website says, it's not my words, it's the largest research center in this field in the nation, and I probably need to say now one of the largest, because I know that other centers are also developing and do similar work, and all of us, we contribute to the field of hemp science. Now at Oregon State, there's 65 faculty involved in the research in 19 academic disciplines, so this is a widespread effort, and they all do research or teaching or involved in extension services. So when we talk about hemp, we need to be clear that it's all one species. It's Cannabis sativa, and there's actually two varieties. There's one that's that doesn't have any cannabinoids, or no cannabinoids that have psychoactive activity. So these are the ones with, as you can see on this slide, less than 0.3% tetrahydrocannabinol. And there's the ones with the high levels of the tetrahydrocannabinol, and that's then the psychoactive cultivars called marijuana. And this is from the famous book, Small and Cronquist from 1976. Now, where does it all start? About 1,000 years ago, people started to use hemp for all variety of purposes. So there's a fiber variety that was used in Europe and also in Eastern Asia. And the narcotic, so the psychoactive varieties, they were grown, and you can see them more in the Central Asia. And again, this is from Small's work in Botanical Reviews in 2015. Now, talking about the fiber hemp, this is the farm where I grew up in the Netherlands, and you can see my father, he grew hemp. You can see this is hemp here next to the farm, and that was all entirely used for fiber. And what did they do with it? Or what do they do with it? They make insulation mats, and also you can see on the right, they mix it with concrete and get this so-called hempcrete, and you can make buildings out of it. Now, what is interesting here is the fiber is, of course, is made out of lignin, which is actually a polyphenol. It's a polymer made of 3 of these phenolic acids, and these make up a very strong network. And is it's the same polymer that is in wood. Of course, hemp grows much faster than wood, and that's why it's very attractive. Now, if you see, the BBC had a report about a month ago, and it was about sustainability of building materials, and there hemp was highlighted as a future material for making sustainable but also very attractive building materials, because they keep the heat out and the warmth inside. Now, this is something from the past. We're all kind of nervous about these psychoactive cannabinoids, but let's not forget about from 1870 to 1942, hemp, and especially the marijuana, because it says here hemp, but this was actually the variety indica, and it was in the pharmacopoeia. And and obviously this was for medicinal purposes. Now that after the Second World War, it was banned from the pharmacopoeia, not only in the United States but also in Europe. You talk about the bioactive compounds. Luckily, they're excreted by trichomes, and why I'm saying luckily, because that makes the extraction a lot easier. So these hemp, they on the leaf material, the green material, you can see there's a little stalk and there's a little cell cluster, and they excrete a very sticky material, and that sticky material is very rich in cannabinoids, but also terpenes that give you the specific flavor. Now, I'm not going to talk about the exact difference between marijuana and hash or what, because most people know that already. Now, there is great variety in these cannabinoids. If you look in the literature, there is more of them. Now, what I'm hearing at the conference that I'm right now, it's about 150 compounds, and they all start with an acyl-CoA, and in this case, I'm giving the example, the C6-CoA, so hexanoyl-CoA, and then plus 3 units of malonyl-CoA gives you the olivotolic acid, and the olivotolic acid combining, it gets phosphoryl, it gets with the geranyl diphosphate, it gets a new CC bond, and then you get the first, say the cannabinoid, cannabigerolic acid, and you can see this prenyl group here, it can make a positive cation, and that can then form cyclizations with neighboring groups, and that gives you rise to a large variety of cannabinoids. Now, the cannabinoids is only one class. From the geranyl diphosphate, you can make monoterpenes. From the farnesyl diphosphate, you can make sesquiterpenes. You can see that the number of natural products in hemp is enormous, and that's also why it's very attractive as a platform for development of pharmacological agents. Now, going back to the intermediate, so here's another example. I lifted out the precursor. So there's 3 enzymes that make, let's say, cannabidiolic acid. Then you got to get the tetracannabinolic acid and the cannabinolic acid, and they make then the compounds that we all know, such as cannabidiol, CBD, THC, and cannabinol. Now, why are we now, so the biosynthesis is, of course, this comes from the hexanyl-CoA. So this carboxyl group is easily lost when you treat plant material with heat or whatever, and that's shown on the on the next slide, because you can make a 6-membered pseudoring, and that pseudoring is promoting the release of carbon dioxide. And that's why it's you get all of these decarboxylated compounds. And of course, that changes the pharmacology enormously, because you decrease the polarity of these compounds. And that can be induced by heating. You can see here smoking, but also distillation can give rise to the loss of the carboxyl group through release of carbon dioxide. Now, at Oregon State, my lab, we work with Columbia Basin Biosciences. This is a company in Eastern Oregon, and the website says they're a very large company. So they grow hemp on their own farms, and that's about 2,300 acres. And then they harvest that once a year, and then they dry this, and that drying takes less than 2 months. And then they start processing the material. And here in the inset, you can see they do extraction, then the centrifuge, you filter, then you evaporate the solvent, and then they distill it. And you can see that distillation is done on a large scale. And you can see on the right side here, the distillation columns, these are really huge. And that can be up to 1,000 liters a day. And from that, these distillates, if they tune the conditions carefully, they get a product that is rich in CBD and also CBG. And those products, they can be, you can get the main compound out by crystallization. Now, that crystal material is then pure, but their supernatant is very useful for us because that has the minor components, and we are interested in the pharmacology of the minor components because the National Institutes of Health, they're not so interested these days in developing drugs from THC because of the dependency. Now here's an example from our own lab. So when we run, let's say, a mother liquor, this is the supernatant from the crystallization process. You can see there is still a lot of the CBD in there, but you can see other peaks in there, and these are the other components that are of great interest to us. And we annotate these compounds because we have a in-house library of mass spectral data for automatic identification of cannabinoids. Now, these are the major components here. Oh, here's the library. So what we do is when we run the LC, this is liquid chromatography in combination with high resolution mass spectrometry. So that means you get a mass for your compound in high accuracy and also precision. So that allows you to go into a database, and includes the one that we have in-house, to determine what is what. Now here you can see on the left side, this is a mass spectrum. On the x-axis, you see the M over Z values. You can see there's 3 peaks. It's still the same compound because this is we call that the monoisotopic peak. And then with these molecules, the population molecules, with 1 carbon C13 is 1 mass unit high, and this is 2 mass units higher. So it's all, we need to be careful that we annotate these compounds carefully. And if we then look at this monoisotopic, we can break it up. It's called MSMS. We make fragments out of it. And the fragment mass spectrum is a fingerprint for the identity of these compounds. Now, if we do this, then the software will tell us, here you've got a single peak for that particular product, and then it identifies this as cannabidiolic acid. That's how we do it. Now, if we apply this method to the minor components, and this is all the major components, then you can easily get a list of compounds that are automatically annotated. You don't have to do it manually anymore because we do all the pre-work by setting up this library. Now, here's another example with the minor component. You can see there is, in addition to the large peak of the CBD, there's many, many minor peaks that are annotated based on the library. Now, what happens if you do not have these compounds in your library? Then, of course, the in-house library will not tell you what a peak is because the mass spec will only generate peaks. Then we go to other methods. And this is an example where we use the so-called Global Natural Product Social Networking tool, the GNPS tool. And it has 2 components. So this is an online tool that's available to any researcher around the world because it was funded by a taxpayer dollar, so it's freely available. And it has 2 components. One, it has a large library. So if you're lucky that there are cannabinoids in there that you don't have in your in-house library, but it's still online available. And of course, there's still a possibility that some of these compounds are not in your own library, also not in the GNPS library. Then we resort to the so-called GNPS networking. So how does this work? So I told you on the previous slide that if you do your mass spec experiment, you get MSMS spectra, the fragmentation spectra. Now, if you assume, let's say that, now I told you about the C5 chain that comes from the hexanyl-CoA. If there's a longer chain, and of course, all these peaks in the fragmentation spectrum will probably, well, some of them will at least by shift by 2 carbon atoms. So that's a corresponding mass shift. And but the appearance is still looks the same. So in this example, with 3 spectra that are related to each other, this and this one, the initial peaks that are a little shifted, and here you can see these are a little shifted, but they're still related to each other because some of them still have a common fragment, and then it aligns, the software will align these components, and it represents each molecular species, so that's a compound in a network. And then you get this so-called network. And then if you, let's say, suppose you know this blue node, so the blue compound, suppose that is in your library, but not the gray ones, then at least you know these gray ones are structurally related to the blue one, and it allows you to do at least an attempt to assign the structure to these gray ones based on the similarity to something that you do know. Now here we have done this with our CBD mother liquor. So that again, that's the supernatant from the crystallization process. And each node, so each of these blue dots, that's a compound that we actually detect by mass spectrometry. You can see the richness of the chemical composition of that purified or that enriched material besides CBD. And if you look, of course, not everything is cannabinoid. There's other terpenes in there that are not cannabinoids. But if you then look at the cannabinoid cluster, so we know some of them in this cluster are cannabinoids, so we call that the cannabinoid cluster. And then this is zoomed in, and you can see if you look carefully, and it's kind of small, but some of them, they have the blue annotation based on our own library, and the CBD is right here. And CBCA is right there. You can see many of these compounds we can already annotate based on the tools that we have in hand, but there's also compounds in between. So especially in the inner core here, they have no annotation. And then it's, of course, the puzzling starts and see what are those compounds, because, of course, that mixture, if you would test that pharmacologically, they clearly contribute to the overall effect in any system that you are using for testing. Now, so here is that, again, the annotation with all of the MSMS spectra. You can see CBD is here, CBG is over there, and then on the other side, CBCA. Now, you get the gist that many we can already annotate, but there's still in the inner core many compounds that we do not know at this point in time. And that's when we resort to looking at what are the connections of these unknowns with the known ones, and I have made these yellow so that we lift them out. And then we discover something, because many of these compounds, they are actually, we call them homologs of CBD. So CBD, as I said before on that slide, with that biosynthesis pathway, they have a C5 in the side chain of that aromatic ring, and that comes because of it starts with the hexanyl-CoA. And 3 of these units of the malonyl-CoA. But we call that the CBD C5, that's the regular CBD. But there's also a CBD with 6 carbon atoms in the side chain, and there is one with 7 carbon atoms in the side chain, somewhere with 8 carbons and 9 carbons, and 11 carbons and 13 carbons. So that means that for each of these components that you find that everybody is annotating, there is probably maybe 10 more of those where the variability comes in, or the chemical diversity is caused by the chain length. And of course, the chain length is important because they add either phobicity to your molecule, but also if you work with receptors, that the longer the chain length, they may either bind better or not as bind as good, and then you lose the activity in the biochemical assay, in a pharmacological assay. Now, why are we doing this? And I don't have the results yet for the mother liquor, but I worked for the last quarter century on hops, and the hops, they have similar compounds because they're from the same family, right? Humulus and cannabis, they all belong to the family of the Cannabinaceae or the Cannabaceae, depending on the literature that you read. So the chemistry is very similar. So what we've done is we make a hop extract. In this particular example, we make a hop extract and we run it through a column, and then we collect fractions. You can see that here. And these fractions are then tested in a bioassay. And this is a assay for measuring nitrite production. And this is a measurement of inducible nitric oxide synthase activity, and that's actually an index of inflammation. Now, in a cell, and we use here mouse macrophages, We stimulate the cells with lipopolysaccharide. That's a breakdown product of bacteria. So that's highly inflammatory. So the inflammation signal will induce the production of the activity of the inos, the inducible nitric oxide synthase, and that process will then give you eventually nitric oxide. When nitric oxide is not stable, it will produce nitrite, and the nitrite we can then measure in an assay. So if you then look for the activity, so of course, if there's a lot of stimulation, you get a lot of nitrite production. So if you have a lot of inhibitory activity of your compounds on this enzyme, you will see that the nitrite production will go down. Now here on the left side, this is the complete extract, so it has some activity. Here's the positive controls. This is about 100%, and this is about, let's say, maybe 60%. You can see that some fractions, they do not contribute to the activity, but there are also fractions, and you know, the fraction 34, for instance, here is a highly inhibitory of that particular activity. Now then the question is, what is it? And then of course you look in your mass spectra that you record at the same time. So you combine the bioassay data with your bioassay data, and then by machine learning, so this is artificial intelligence, you can actually make a rank list of compounds or masses that the software predicts to be contributing to the activity most. Now, then you can see here there's a top list, and of course you can go down the list as far as you want, but the top list is, so there's a compound with 353 in negative ion mode, there's one at 355 and 369, and let's assume for the moment that we don't know what that is. So then we go to the next step, and this is then the example of the GNPS networking that I talked about. And then it so happens that, so this is a cluster here where we find xanthomyol. So we know that this compound is actually, and that it was coming from the database, that is the most active compound in this assay. But of course, we knew the other ones we don't know, but they still show a structural similarity with nodes that are connected by the MSMS spectra similarities. And then you can identify the other ones as well without actually having these standards in hand. Now, there's still predictions. So now we can go back to the data, and then we come, oh, sorry. So this is, so here, then we can annotate the top predictors of activity as xanthomyol. Then there's a compound called dihydroxanthomyol and xanthomyol B. And of course, these are still predictors, so it's not said that these actually do the work. So you still need to validate the results. So what we do then is we take pure xanthomyol that we isolate from the plant material, and then you run a curve, a pharmacological S curve, and then you compare that with the fractions with the same amount of that compound in those fractions. And you can see the black one is the pure xanthomyol, and the blue ones are the fractions that lead up to 34, and the ones that tail after the 34. You can see the fractions 30 to 34, they are shifted a little bit to the left, so that means that there is something in there that helps with the activity of the pure compound, whereas the late fractions, there is something in there that is actually pro-inflammatory. And then, of course, you can actually look again at your data and look for masses that are predictive of the inhibition. And actually, if you read the paper, I don't have the time to talk about it today, but we actually identified the pro-inflammatory compound in those fractions as well. Now, so far as the chemistry, and now we have, I've talked about how to get these compounds and how to make predictions of activity. But if you talk about the pharmacology, everybody is, of course, interested in, or at least the NIH is interested in pain relief, because for chronic pain, there is actually very few therapeutic avenues for treating chronic pain. Now, everybody is looking at the cannabinoid receptors. There is, of course, there is the CB1 receptor that's highly distributed and very abundant in the central nervous system. And they are contribute to pain relief, but also they're also responsible for the the psychoactive activity, and that's not good because that induces the addiction to this compound, and that is not desired. Now, there's also a CB2 receptor, and the TB2 receptor is expressed mostly in the periphery on immune cells. Now, besides, oh, I forgot to mention, so when we talk about cannabinoid receptors, then the question I often get is, why is there a cannabinoid receptor? Because did the humans make cannabinoid receptors for cannabinoids? No, of course not. These cannabinoid receptors, they were already long before humans started to expose themselves to cannabinoids. Now, it so happens, and that is the same is true for, let's say, the opioid receptor. There's always an endogenous ligand, and the endogenous ligand has a molecular shape that is similar to the drug. And that's also for any system in pharmacology. So here's, for instance, this is THC, this is the drug, this is the molecular shape. So there is an endogenous ligand, this is called anandamide, and that is a amide from arachidonic acid, and the model says that it has a shape that it fits very well to that receptor. Now, what is also interesting, that the original discoverer of THC, we all know that's Professor McCullum, but he also actually identified the endogenous ligand for these receptors, so that's quite remarkable. Now, when we talk about pharmacological applications, there is some of them have already been approved by the FDA. And examples are for THC is dronabinol and marinol, and the synthetic analog is nabilone. And those are approved for inhibition of chemotherapy-induced nausea and vomiting. There's also appetite stimulation and pain inhibition. Dronabinol is approved for some of these applications. There's inhibition of tumor growth that's still very much in the experimental stage. Now then, cannabinoids, unlike what many people believe, they have actually very favorable drug safety profiles and do not produce toxic effects in conventional chemotherapies. Now then, there is other applications such as some of them have been experimented for treatment of Tourette syndrome. And there is a mouse spray that's approved in Canada and Europe for multiple sclerosis. Now, what is interesting here, that if you say, okay, stimulation of the CB1 receptor will increase appetite, so that's for the, let's say, the people that terminal cancer patients that have no appetite to stimulate their metabolism. But you can also turn it around and say, can we develop something, a drug from this platform that actually antagonize the receptor or actually inverse the activity of the receptor? And the pharmaceutical industry has done that and came up with the compound here, rimonabant. And that was actually used in Europe for a while as a compound for weight loss. So it was approved in 56 countries of severe obesity and not in the US. But then a couple months later, in October 2008, it was withdrawn from the market due to side effects, including depression and suicidal thoughts. So that was not so good news. But nevertheless, you can see that you can use the cannabinoid platform to develop very valuable drugs. Here's another example. These folks that published this paper, they have taken the CBDA, you can see as the carboxyl group, and they have esterified the carboxyl group with the methanol, and then you get a compound they call EPM 303, and they tested this in a mouse model of obesity, and they find, let's say, if this is the vehicle, so this is just the normal diet, and then you give the animals a high-fat diet, you can see that the weight of these animals increases, and if you then treat these high-fat-fed animals with this compound, then you can see there is a reduction in body weight. Now, that is significant here in this paper. If you think, no, that's not so impressive, let's not forget, this is more than 10% weight reduction. And if that would happen in humans, that would be fantastic news. Now, they also looked at fat mass is reduced and the activity here. So this is the wheel running. This is a measure of how active these animals are. On a high fat diet, they're not really active at all, but if you give them then this compound that's derived from the CBDA, then the activity is back to normal levels. So this is all very encouraging news. If you didn't make slices of the liver, and they did that, so this is what you see on the middle one with this high fat diet, you see these hepatocytes. So these are the liver cells. They have all these droplets and this full of fat, and that's not good because that will induce liver fat and also this non-alcoholic fatty liver disease. And that can eventually cause, cirrhosis, so that's not good. But if you didn't treat with the compound at the same time, the livers, they look like animals that were on their normal diet. Now, this paper does not talk about the mechanism of action, but I have done similar work with my hops compounds, and I brought it and said, well, maybe the mechanism of action is through activity on the mitochondria. Now, mitochondria are little organelles in your cells, and they generate ATP, and they're also responsible, of course, for oxidation of fat as a source of biofuel. Now, what we do is, in the lab, what we do is we looked at the membrane potential in these mitochondria, and if the membrane potential is lower, that means that the cells, they need to work harder to produce the same number of ATP molecules. And if in that process, of course, you burn more fat. So that is good for people with obesity. So for instance, here in the ethanol control, there's normal aggregates going on. So the delta xi, that's the intermembrane potential, that is a measure of the mitochondrial activity, is normal. And here in the THC 13 micromolar, you can see that the mitochondrial membrane potential is lower, and we'll explain in the next slide how that works. You can see here's the quantitation. So here's the normal one with the vehicle control, and here's 8 micromolar, 32 micromolar of THC. You can see that the membrane potential is greatly reduced. Now, going to the next slide, here is the model that we published in 2018. So here we talk about this mitochondrial uncoupling that affects the membrane potential. That's the delta xi here that goes down, and the cells respond by producing heat and less ATP production, or more ATP, and then that affects the so-called adenylate energy charge. And if that is reduced, then the cells, they get a signal to produce more energy by burning more fat. And that maybe that is a good explanation for the folks that published that paper. Now, going to pain, here's a little scheme of where pain comes from. So here's a pain. So here is a neuron. This is called a primary afferent fiber terminal. And when you get a pain stimulus, it releases glutamate. That's the neurotransmitter. And the neurotransmitter is then entering the dorsal horn neuron, and it causes pain. So there you get the pain signal. But of course, there must be a antagonistic physiological system to actually reduce the pain. And for that, there is 2 neurotransmitters, and one of them is glycine. And glycine is transported to that dorsal horn neuron through the so-called glycine receptor. Now, if the glycine, that is an inhibitory neurotransmitter, if that is transported across the cell membrane through this receptor, then that will cause a reduction of the pain signal. Now, of course, for that, there is compounds in there. These are from the endoprostanoid pathway that actually inhibit this pathway. So if you could inhibit this pathway, or you could stimulate this pathway, that would be a way to decrease pain signal. And that's independent of the cannabinoid receptor. So of course, this is highly desirable to look into further, because that is not causing the dependency on these compounds. So here is a little summary of that paper that I reviewed. Here's the glycine. You can see that the more CBD is added to this system, the more signal, the more activity of the glycine is in there. There's more glycine signaling. So that means that this is really a mechanistic action that can be further exploited to use the glycine receptor as a target for reducing pain. Now, there's another receptor system and that's the so-called, you can say there's a long name, the transient receptor potential cation channel subdomain 5, member 1, so the TRPV1. Now, we call it the capsaicin receptor. We all know capsaicin, that is a very pungent compound, and it causes pain when you rub it on your skin, for instance, after you have, or on your muscle, you have a sports injury, and that pain is actually considered to be a more tolerable pain than the pain that comes from the injury. But nevertheless, these receptors, they are involved in pain signaling. Now, what is here, the mechanism here, you can see this is again a pharmacological curve. So this is on the x-axis, you've got the concentration of the cannabinoid that they tested, and here's a whole list of them that they in this paper. And on the y-axis is the total pain signal, but of course you first need to stimulate that system by adding capsaicin to get a pain. And then you look, can the cannabinoids reduce the pain? You can see CBD is a fairly potent inhibitor of that pain signaling. And then the ones that the carboxylic acid is a weak desensitizer of that pain signaling. But nevertheless, you can see that these compounds have really affect the activity of the capsaicin receptor pain signaling. And that can also be further exploited. As an interesting detail is that in 2021, actually the Nobel Prize for the discovery of the capsaicin receptor went to Julius and Pataputian. Now, we have also done our work on THC, actually, together with folks at the Oregon Health Sciences University, and they have access to the primate center in Portland. And our primate center, we use macaque monkeys. And the idea here was, if you take, let's say, blood platelets, and you stimulate them with endocannabinoids, and like anandamide, these platelets, they start to aggregate. Now, that is cause for concern, because if the cannabinoids would do the same thing, they would cause blood clotting. And of course, people that are taking medicinal THC, that will be a very bad side effect. So there was concern, and we tested if that is happening with THC itself. So what we did is we took monkeys, and there were males and females, and then we gave increasingly a higher dose of the cannabinoid. You can see here is the dose is up here, and the blood draws. And then what we did is, at the end of the study, we looked at, does that compound THC have an effect on blood clotting? And you can see here is this is the collagen-coated microfluidic chamber, and then you look how much of the platelets stick to the collagen if you blood flow through the system. You can see at baseline there's a lot of clotting going on. But if you then treat with THC, and this is blood from these monkeys that we spin down, and you get the platelet-rich plasma, you can see there is, of course, in this cartoon, there is much less aggregation of these platelets going on. And if you then quantify that, here's a micrographs of that. So at the baseline with 0 milligrams, then there's a lot of clotting, but at the, let's say, 2.5 milligrams per 7 kilograms, so that's the monkey weight on average, there is much, much less aggregation going on, and that's good news. And here's the quantitation. You can see that there is a dose-dependent inhibition of platelet coagulation going on. Now, we looked a little bit at the mechanism. And the mechanism is here, if you use a blood platelet, and how does the aggregation is stimulated. Now, there's so-called glycoprotein 6 receptor, and that's stimulated by the so-called collagen-related peptide. Now, that is stimulating the aggregation. But if we then first stimulate these cells with this collagen-related protein, and then at the same time take blood from animals that were treated with THC, you can see again a dose-dependent reduction in the platelet aggregation. That's over here. So that at the highest dose, there's much less aggregation going on. And if you measure the aggregation by light transmittance, you can see that the highest dose, there's much less aggregation going on. Now, that was the last slide that I wanted to show. I see that it's now 1:45. Unless there is desire for me to read out all the conclusions, but I think that well, the conclusion here is I think maybe the last thing is pretty important for the future of this work. So there is large chemical diversity in the cannabinoids, and everybody looks, let's say, at THC or CBD, but nobody looks at the other compounds and how the other compounds in the hemp extract will actually determine the overall effect of on a, let's say, a pharmacological system. The example that I gave with endocannabinoids and the phytocannabinoids, they have really opposing effects. So it's not always in every system that they give the same results, and therefore we need to really carefully tease out what is doing what. And for that, we need chemistry to separate these compounds, determine the structure of these compounds, and then either through artificial intelligence system to make prediction to save time, because at least you know what to look for. Actually, in the end, all of the compounds need to be carefully estimated what their contribution is to the whole pharmacological effect. And that was my conclusion, or at least that's how I see the future. And now I'm Dr. Bernstein, are you there for taking questions?
Hi, Professor Stevens. Can you hear me?
I can. Do you want me to stop sharing?
No, that's fine. Whatever you want, it's fine.
Okay, I'll stop
We have questions. We have questions, of course. First question is for the chemistry part of your talk. And again, thank you very much for a very enlightening and interesting talk, and for summarizing, you know, all those this topic and giving us an overall overview with an in-depth look on current studies. So that was very nice. Let's start with the question. One question is for the chemistry part of the talk. In light of the many small cannabinoids that we found recently, can you speculate, or do we have any information about what we should expect for in the future? I guess what the future is holding in terms of advances in cannabis chemistry and potentially more cannabinoids or minor cannabinoids to be found.
Well, what is it? Results from the past are no guarantee for the future, right? So if you look at my own hop research, I found, for instance, if let's say I got the xanthomyl, that is a molecule with C5 in the side chain, if I extend that chain length by another 5 carbon atoms, that greatly enhances the anti-inflammatory effect. So if you extrapolate that result to the cannabinoids, then I would be greatly interested as in this CBD, not with 5 carbon atoms, but let's say with 11 or 13 carbon atoms, because that would, if they work through the same pharmacological mechanisms, they would be expected to have enhanced effects. And of course, when you know that, they don't need necessarily to come from hemp, right? If you have a better way of synthesizing those molecules, there will be another source. But at some point, we need to see what is the chemical diversity and use that chemical diversity to come up with hypotheses of pharmacologically very strong compounds, or very potent compounds.
Thank you. Another question for the chemistry part of the talk is that, as we know, there are cultivars, there are many differences and variants in the chemical profile between cultivars. Do we know at this stage of time anything about what is the source of the differences in the biochemical control of these pathways which cause this diversity?
I think there's not so much known. Yeah, what can I say? What everybody would say, maybe environmental influences. Yeah, you can have mutants where they're less restrictive. Let's say for the first enzyme that appears to be liking the hexanoyl-CoA more than the others, or that actually take only 3 malonyl-CoA units. Maybe the other ones that are less specific, they may make a larger variety of compounds, and they would, of course, also that would affect the pharmacology. So it's difficult to say what is the cause, and I think that's also interesting because I said these fractions that I made in the lab with one extract, you could do the same experiment with different cultivars as long as there is variability between these specific samples. So this is all things I think that still needs to be done before we before we all focus on a single compound. I think that's not the right approach.
Yeah, I think also, thank you for this answer. And I think that you've said, I mean, that we know so little even about the biochemical pathway that, you know, we're really wishing to get to soon to the position that we can work about regulation of the pathway.
And of course, that's going on as well.
It's coming, you know, and probably soon.
But this would be very good to those folks that do, you know, I'm here at the moment, I'm at a bioengineering conference for plants. And yeah, those folks, they know how to engineer pathways, but what do you really engineer? I think you need to know what you look for and what you want. Just to make a compound is one thing, but if it's not useful for any application, then it will not lead to anywhere that we can use in the future. So I think there needs to be collaboration between those folks, the chemists and the pharmacologists, to actually find what is actually useful to develop further. Because if we don't make any progress, then the hemp hype will be soon over and people will resort to old-fashioned chemistry again, or synthetic chemistry, which is what we don't want.
Right. So we highlighted again, you know, the incredible lack of knowledge in terms of medicine, pharmacology, plant biology, plant biochemistry, and whatever. There's still a lot needs to be done, and as you said, preferably quickly.
Yeah, no, I learned myself in this project, the GMPS network. It was a fun tool to work with, but seeing how powerful this is to actually highlight what compounds are in there that are actually related to compounds that you do know. Because let's not forget, if you get a mass spectrum or you get LCMS traces, there are probably hundreds, if not thousands of peaks in there. And these peaks, you don't know what they are. So only relying on an in-house database, that's also not sufficient. You need to have also discovery tools. And that's why I'm pushing the discovery much to make new things and discover new things, and also in terms of pharmacology.
We look forward to hearing, you know, breakthroughs and new information. Another question is, most cannabinoids are produced from CBGA, but the minor cannabinoid pathway is also through the CBGVA. Would you happen to know, or if we have any information or any research specific, either medical or pharmacological studies which are conducted on the activities of this minor pathways, cannabinoid in those this minor pathway?
Oh, I don't know the answer, but I totally love this question, and I tell you why. So the National Institutes of Health, they came out with a call for proposals, and they asked specifically for minor components from different different biosynthetic pathways to look at the pharmacology. So apparently, people know kind of today what THC is doing, what CBD is doing, but they have no clue what the other compounds are doing. And that still the science field needs to figure out. And actually the NIH is allocating money to do this. Unfortunately, I don't have the answer, but we work on it.
Nice. Yeah, another related question is, have you done any work with cannabivarin?
I have not.
So,
not as a, no.
Yes, and again to our audience, you know, the V in the CBGVA is the varin part of this, of course. Another question. Thank you for your excellent presentation. In your HOP anti-inflammatory study, how do you prevent nitrite from becoming nitrate? Are you concerned you may be underestimating the effect through loss of some of the nitrites?
Okay, well, you know, this is the so-called grease assay, and that is generally accepted in the field as a measurement of INOS activity. So of course I know fully well, and I agree with the whoever asked that question, that it has limitations. But actually it is a very cheap assay. So if you want to do a first screen of compounds, this is actually a good starting point. But of course, later on, you need to do the hard work and then also look, for instance, at compounds that are, let's say, the inflammatory cytokines. And we have multiple papers on the hop extract and also hop compounds where it affects, let's say, IL-6 production. And we actually done it preclinically in animal models of inflammation, where we actually showed that this case of the xanthomol, that we can actually reduce the circulating amount of IL-6, which is a pro-inflammatory compound, by 80%. So this is all things that this is the first start of a bioassay, but then you need to follow it up with other things. You cannot rely on a single assay and make very bold conclusions. Well, I've been long enough in the field to realize this. And actually, for the audience there, the person who asked that question, so we followed up on the IL-6 story, and actually now we have our hops compound in clinical trials for inflammatory bowel disease treatment together with folks in Portland. So you can see that a bioassay in cells, then you go to an animal model of inflammation, and then now we test this in humans. And the fact that the NIH believes in this tells you that we are on the right path here.
Thank you. Another question is, in the CBDA-derived obesity study, what would be the equivalent human dose of CBDA, which is required to produce similar results? In the mouse dose, about 40 milligram per kilogram per day, it appeared to be very large.
Well, I'm glad that person asked that, because there is a thing called interspecies scaling of dose. And the relationship between humans and mice is 12. So that would be, if you divide 40 by 12, what is it? 8 or what is it? Yeah, less than 10. It would be less than 10 milligrams per kilogram. So it is not as high as in mice, because they have a different shape of their bodies. So we need to divide that dose by 12. So then it would be, now if I would, let's say, suppose it is 5 milligrams, right? Suppose. Then it would be 350 milligrams in a 70 kilogram person, if I roughly scale it that way. You can certainly not compare mice and humans on body weight. That's not how it works. And actually, we have done this interspecies scaling of dose with our hops compound, and actually works very well, because we could reproduce the effects, the pharmacokinetics of the hops compounds in rats, actually, with humans, and that scaling works perfectly well if you look at the body surface and not the body weight. And the FDA has a protocol for this in drug development.
Okay. Thank you.
But if I may add to this, if you look at the 2.5 milligrams of THC in that monkey model, so this was 7 kilograms, so they weigh about 7 kilograms. So they are a very small dose, will actually have profound pharmacological effects on platelet aggregation or inhibition of platelet aggregation. So that's very encouraging. It's not that like these compounds don't do anything. They're really potent.
So, and as you can say, as you said, we can compare, between dosages for mice, and probably dosages for humans. It's interesting. Another question is, have you observed that synthetic cannabinoids show different results in contrast with non-synthetic cannabinoids? Are there any experiences or any studies about this?
If it's the same compound, it shouldn't matter where they come from. As we have done synthetic xanthomol, and it behaves the same, because if it's chemically the same, they have the same pharmacological effect. It does not need to be from a natural source. And the same is true when I talk to my students in class. The same is true for all drugs and all vitamins. Vitamins always ask, is synthetic vitamin C better or worse than natural vitamin C? Well, it's the same molecule, so it doesn't matter.
Mhm. And we're already past the hour, but we have many, many questions. We'll take one last question in
Okay.
order that it was written. And then about pain research. Do we know of any interactions between cannabinoids and terpenes in pain reduction?
I don't think there is, but again, I point to the call for proposal from the NIH. They are really interested in what is it, interaction between cannabinoids and other compounds in hemp. And actually, the model that I described, the fractions, remember, what we can do is we can take one fraction, which is potent, and then add aliquots of other components, including terpenoids, to those and look at, again, at the bioactivity. So this is all research that still needs to be done. NIH is really interested in this. I'm really interested in this, so we just need to go to the lab and do the work.
You know, again, a lot of questions and unknowns. You know, we have to start from someone and progress forward. So again, Professor Stevens, thank you so much for sharing, you know, your information and background and studies from your lab. And we really appreciate, you know, the time spent on this to join us for this webinar. And have a very nice continuation of the day. Thank you.
Oh, thank you very much. And everybody have a good day. Bye-bye.
Have a good day, everyone. And please join us for our next week's seminar as well, webinar. Bye-bye.